everyone, and welcome to the video. My name is Lori, and today I'll be talking about how cells get and use energy, including metabolism, enzymes, the catabolism of carbohydrates, cellular respiration, and even fermentation. So let's start out by explaining metabolism. You may have heard of the term metabolism in reference to weight loss or weight gain. For instance, someone might be able to eat an entire family-sized bag of chips and stay thin. And you would say, well, they just have a great metabolism. But what does that really mean? Metabolism refers to all of the chemical reactions inside of cells. This includes catabolic reactions that break down large molecules like starches into simpler sugars, and also anabolic reactions that build up simple molecules into bigger ones. These two things happen in tandem. Breaking down molecules releases energy to fuel the processes that build up molecules. Metabolic processes require carbon because life, as we know it, is carbon-based, and we can identify two types of life based on how they gain carbon for these metabolic pathways. First, there are autotrophs, like plants, which convert inorganic carbon dioxide into organic carbon. And then there are heterotrophs, like humans, who obtain complex organic carbon, mostly from autotrophs. Prokaryotes are also heterotrophs. As we mentioned earlier, larger molecules are broken down to make smaller molecules, which releases energy for all these metabolic processes. The energy is held within the bonds of these larger molecules in the form of electrons. Electrons have a negative charge, so when a molecule accepts an electron, it's considered to be reduced. When a molecule loses its electron, however, it is oxidized. This usually happens at the same time, and so they're called a redox reaction. When redox reactions happen in living cells, molecules that are easily oxidized or reduced are called electron carriers. These electron carriers include NAD, NADP, and FAD. They serve an important role in extracting energy and carrying it to be used in chemical reactions. All this energy is great for running the cellular processes, but cells need to be able to store the energy somehow. That's where adenosine triphosphate comes in. This molecule stores loads of energy in its unstable phosphate bonds. That can be easily broken to release energy, making adenosine diphosphate, or even one step further, adenosine monophosphate. Because it can store energy, the cells can use the energy when it needs it, instead of all at once when it doesn't. All of these chemical reactions and breaking down of nutrients also require energy for them to occur, called activation energy. To speed up some reactions, the body uses enzymes to lower activation energy. Most people have heard of lactose intolerance, so let's use this as an example of how enzymes work. To break down the main sugar of dairy products, lactose, the body uses the enzyme lactase. Lactase reduces the activation energy of breaking down and makes it possible for our body to break down lactose, so we don't get a stomach ache when we say drink a milkshake. Basically, enzymes have an active site that binds and conforms to a specific substrate, like lactose, and it either helps break it down or combine it with another substrate. In the case of lactose, it's broken down into galactose and glucose. Enzymes often need help from cofactors or coenzymes to do their job. Cofactors stabilize enzymes, and they are inorganic, whereas coenzymes actually activate the enzymes by binding to them. Neither enzymes nor coenzymes are used up in these chemical reactions, so they can move on to be used again in later reactions. Once the body has glucose, it's broken down to create ATP and the reduced form of NAD plus NADH in a process called glycolysis. The most common glycolysis pathway is the EMP pathway. Like activation energy, glycolysis also requires energy to begin in the form of 2-ATP. These ATP make it to where the glucose can be broken down into 2 even 3-carbon molecules called glyceraldehyde 3-phosphate, G3B. After this energy has been expended, the G3P can be oxidized, which replaces the first 2 ATP, creates another 2 ATP, reduces NAD plus to NADH with electrons from glucose, and creates 2 pyruvate molecules. However, the breaking down doesn't stop here. Pyruvate can enter another pathway called the Krebs cycle after being broken down in a transition reaction to 2 carbon acetyls by an enzyme and attached to a coenzyme for transport. In prokaryotes, the Krebs cycle and the transition reaction both occur in the cytoplasm. In eukaryotes, like humans, they take place in the mitochondrial matrix of the cell. The Krebs cycle is an 8-step closed process of chemical reactions that create different molecules called intermediates until finally regenerating the molecule it uses in its first step. One of the intermediates that it creates is citric acid, which is why another name for this pathway is the citric acid cycle. After one cycle of reactions, the Krebs cycle has produced 2 CO2, an ATP or similar GTP, 3 NADH, and 1 FADH2, the reduced form of FAD+. So far, all of the ATP that I've discussed has been created through substrate-level phosphorylation, where a phosphate group is taken from an organic molecule and added directly to an ADP to form that high-energy phosphate bond we spoke about earlier. However, most ATP is formed using oxidative phosphorylation. Through cellular respiration, the NADH and FADH2 created so far transfer their electrons through a series of reactions until they reach a final inorganic electron acceptor, either oxygen in aerobic respiration or a different inorganic molecule in anaerobic respiration. These reactions happen in the inner cell of membranes for prokaryotes or in special proteins of the inner mitochondrial membrane for eukaryotes. The energy taken from these reactions is stored in hydrogen ions that are pumped across the membranes, creating an uneven distribution or electrochemical gradient called proton motive force. Since the hydrogen is pumped into concentrated areas, it will want to flow back into the other side of the membranes where it's less concentrated in a process called chemiosmosis. It must travel through a channel, in this case an enzyme called ATP synthase. The flow of the hydrogen ions through this enzyme powers it to phosphorylate ADP into ATP. This hydrogen pumping electrochemical gradient pathway is called oxidative phosphorylation. It can create up to 38 molecules of ATP per glucose, but more often it ranges from 1 all the way to 34. However, not all cells can perform respiration. Cells that can perform cellular respiration get all their ATP from glycolysis, so they need to be able to regenerate their NAD+ from the reduced NADH. To do this, they undergo fermentation. It doesn't use oxygen, similarly to anaerobic respiration, but instead of using an inorganic final electron acceptor, the cell must use an organic one, usually pyruvate. In that case, pyruvate oxidizes the NADH by accepting its electron, creating NAD+ and lactic acid, That's why it's called lactic acid fermentation. Many bacteria that perform this type of acidic fermentation are used in the food industry for making yogurt and cheese from milk. Related fermentation processes are used to make products ranging from alcohol to bread all the way to vaccines. Today we discussed fermentation, the similarities and differences of aerobic and anaerobic respiration, chemiosmosis and proton motive force, substrate level versus oxidative phosphorylation, the electron transport system, the Krebs cycle, glycolysis, enzymes, why ATP, FAD, NAD+, and NADP+ are important, redox reactions, auto and heterotrophs, and of course, metabolism. Thank you all for joining me, and I hope you enjoyed the video. I'll see you next time.